See topic sexual reproduction in flowering plants. Flower, it is a reproductive part of a plant. Male reproductive part is stamen. Female reproductive part is carpel. Stamen consists of two parts, filament and anther. Filament is a long slender stalk and anther is a terminal, generally bilobed structure. The proximal end of the filament is attached to the thalamus or the petal of the flower. The number and length of stamens are variable in flowers of different species. An angiosperm anther is bilobed, with each lobe having two theca. So they are called as dithecous. Bilobed nature of an anther is very distinct in the TS of an anther. The anther is four-sided, that is called tetragonal structure. The microsporangia develop further and become pollen sacs. They extend longitudinally all through the length of an anther and are packed with pollen grains. Next, we are going to see the structure of microsporangium. In a TS, a typical microsporangium appears near circular in outline. It is generally surrounded by four wall layers: the epidermis, endothecium, middle layers, and the tapetum. The outer three layers perform the function of protection and help in dehiscence of anther to release the pollen. The innermost wall layer is the tapetum. It nourishes the developing pollen grains. Cells of the tapetum possesses dense cytoplasm and generally have more than one nucleus. When the anther is young, a group of compactly arranged homogeneous cells called the sporogenous tissue. It occupies the center of each sporangium. Each cell of the sporogenous tissue is capable of giving rise to a microspore tetrad. Each one is a potential pollen or microspore mother cell. The process of formation of microspores from a pollen mother cell through meiosis is called microsporogenesis. The microspores, as they are formed, are arranged in a cluster of four cells that is called microspore tetrad. As the anthers mature and dehydrate, the microspores dissociate from each other and develop into pollen grains. Inside each microsporangium, several thousands of microspores or pollen grains are formed that are released with dehiscence of anther. Now, we are going to see the structure of pollen grain. The pollen grains represent the male gametophyte. The pollen grains are generally spherical, measuring about 25 to 50 micrometer in diameter. It has a prominent two-layered wall. The outer hard layer called exine is made up of sporopolanin, which is one of the most resistant organic material. It can withstand high temperature and strong acid and alkali. No enzyme that degrades sporopolanin is so far known. Pollen grain exine has prominent aperture called germ pores, where sporopolanin is absent. Pollen grains are well preserved as fossils because of the presence of sporopolanin. Exine exhibits a fascinating array of patterns and designs. Inner wall of the pollen grain is called the intine. It is a thin continuous layer made up of cellulose and pectin. The cytoplasm of pollen grain is surrounded by a plasma membrane. When the pollen grain is matured, it contains two cells, the vegetative and generative cell. The vegetative cell is bigger, has abundant food reserve and a large irregularly shaped nucleus. The generative cell is small and floats in the cytoplasm of the vegetative cell. It is spindle-shaped with dense cytoplasm and a nucleus. In over 60% of angiosperms, pollen grains shed at this stage, that is two-cell stage. In the remaining species, the generative cell divides mitotically to give rise to the two male gamete before pollen grains are shed. That stage we can call it as three-cell stage. Pollen grains of many species cause several allergies and the chronic respiratory disorder like asthma and bronchitis. It may be mentioned that Parthenium or carrot grass that came into India as the contaminant with imported wheat has become ubiquitous in occurrence and causes pollen allergy. Pollen grains are rich in nutrients. It has become a fashion in recent years to use pollen tablets as food supplements. In Western countries, a large number of pollen products in the form of tablets and syrups are available in the market. Pollen consumption has been claimed to increase the performance of athletes and race horses. Once they shed, pollen grains have to land on the stigma before they lose viability if they have to bring about fertilization. The period for which the pollen grains remain viable is highly variable and some extent depends on the prevailing temperature and humidity. In some cereals such as rice and wheat, pollen grains lose viability within 30 minutes of their release, and in some members of Rosaceae, Leguminosae, and Solanaceae, they maintain viability for months. It is possible to store pollen grains of a large number of the species for years in liquid nitrogen, that is minus 196 degrees Celsius. Such stored pollen can be used as pollen banks, similar to seed banks in crop breeding programs. Next, we are going to see the structure of pistil or carpel, or we can call it as gynoecium. The gynoecium represents the female reproductive part of a flower. The gynoecium may consist of a single pistil. In that case, we can call it as monocarpellary. Or it may be more than one pistil. We can call that as a multicarpellary. When there are more than one, the pistil may be fused together. In that condition, we can give the term syncarpous or maybe free, apocarpous. Each pistil has three parts: the stigma, style, and ovary. The stigma serves as a landing platform for pollen grains. The style is the elongated, slender part beneath the stigma. The basal bulged part of the pistil is the ovary. Inside the ovary is the ovarian cavity. We can call it as locule. The placenta is located inside the ovarian cavity. Inside the ovary, ovules are present. The number of ovules in an ovary may be one or many. The megasporangium, that is ovule. Let us familiarize ourselves with the structure of typical angiosperm ovule. The ovule is the small structure attached to the placenta by means of a stalk called funicle. The body of the ovule fuses with funicle in the region called hilum. Thus, hilum represents the junction between ovule and funicle. Each ovule has one or two protective envelopes called integuments. The integuments encircle the nucellus except at the tip, where the small opening called micropyle is organized. Opposite the micropylar end is the chalaza, representing the basal part of the ovule. Enclosed within the integuments is a mass of cells called the nucellus. The cells of the nucellus have abundant reserve food materials. Located in the nucellus is the embryo sac or female gametophyte. An ovule generally has a single embryo sac formed from a megaspore. Now, we are going to see the megasporogenesis. The process of formation of megaspore from the megaspore mother cell is called megasporogenesis. Ovules generally differentiate a single megaspore mother cell in the micropylar region of the nucellus. It is a large cell containing dense cytoplasm and a prominent nucleus. The megaspore mother cell undergoes meiotic division. Meiosis results in the production of 4 megaspores. Of the 4 megaspores, one of the megaspore is functional, while the other 3 degenerate. Only the functional megaspore develops into the female gametophyte. This method of embryo sac formation from a single megaspore is termed as monosporic development. Let us study the formation of embryo sac in a little more detail. The nucleus of the functional megaspore divide mitotically to form 2 nuclei, which move to the opposite poles, forming the 2 nucleate embryo sac. The 2 more sequential mitotic nuclear divisions results in the formation of the 4 nucleate and later the 8 nucleate stages of the embryo sac. It is of interest to note that these mitotic divisions are strictly free nuclear, that is, The nuclear divisions are not followed immediately by cell wall formation. After the 8 nucleate stage, cell walls are laid down, leading to the organization of the typical female gametophyte or embryo sac. 6 of the 8 nuclei are surrounded by cell walls and organized into cells. The remaining 2 nuclei, called polar nuclei, are situated below the egg apparatus in the large central cell. There is a characteristic distribution of the cells within the embryo sac. 3 cells are grouped together at the micropylar end and constitute the egg apparatus. The egg apparatus in turn consists of 2 synergids and 1 egg cell. The synergids have the special cellular thickenings at the micropylar tip called filiform apparatus, which play an important role in guiding the pollen tubes into the synergid. 3 cells are at the chalazal end and are called the antipodals. The large central cell, as mentioned earlier, has 2 polar nuclei. Thus, the typical angiosperm embryo sac at maturity, though 8 nucleate, it is called as 7-celled.